All right, so thanks everyone for coming. Welcome to the monthly New York State IPM academic seminar series, in which we host to increase awareness of new research and techniques that advance IPM and its adoption in all types of pest management settings. This monthly series continues through April. Next month, we'll have Justin Renkema talking about insect pest management in fruit crops. Check out the New York State IPM website for more details. For those of you on Zoom, we've got 19 folks on Zoom currently, and probably more joining soon. We're broadcasting today from Cornell's Ithaca campus, which is located on the homelands of the Gayokono, part of the Haudenosaunee Confederacy, an alliance of 6 sovereign nations with historic and contemporary presence on this land. The Confederacy precedes the establishment of Cornell University, New York State, and the United States of America. And we acknowledge the painful history of Gayokono dispossession and honor the ongoing connection of Gayokono people, past and present, to these lands and waters. So today we're very lucky to have Caio Bernarro. I always want to check before saying someone's name if I say it right. Did I say it? Okay. Caio Bernarro is an assistant professor in the Department of Plant Science at Penn State. The goal of his program is to generate basic and applied knowledge to minimize the impacts of invasive plant species in agriculture in a world threatened by climate change and food insecurity. To address these challenges, they focus on 2 areas of research: identification of traits that confer invasive plant species resilience to climate change, and elucidation of how invasive plant species adapt to agricultural practices. His program uses basic insights to better inform management decisions. So thank you everyone for coming, and the floor is yours, Caio. Thank you.
Thank you, Brian, for that introduction. First of all, thank you so much for the invitation to be here today. It's my pleasure. Let me see, how can I pass the slides? Okay, it's working. I might just use the pointer. So what I wanted to do today is to talk a little bit about my research program and some of the things that we are doing to try to elucidate herbicide resistance in agriculture. I'm showing here a picture of Italian ryegrass. It's a plant, it's a picture taken in wheat. It's infesting wheat in Oregon. And this is one of the species that I work the most. And then I'm going to show you later how this can be a very good species to use as a model to try to understand herbicide resistance. So for the presentation today, I will start trying to put everybody on the same page in terms of herbicide resistance around the world and in the United States, discuss a little bit of the physiology, more of the dynamics of herbicide resistance, how it works, the ways that weeds can evolve resistance, and then focus more on annual ryegrass. Sometimes we call annual ryegrass Italian ryegrass. It's the same species, depending on who you talk to, because, for a number of reasons. So, for example, in Oregon, it's a state that produces a lot of annual ryegrass grown for cover crop. So there is a little bit of reluctancy to call them Italian ryegrass. They like to differentiate Italian ryegrass, talking about the weed, and annual ryegrass, the cover crop, even though they are the same species. So again, this is a good model to understand resistance, and then some of the challenges and opportunities to understand herbicide resistance in terms of biology and the management. So weeds are super important in agriculture. They can compete with crops. They can reduce yield in a number of different cropping systems. This is a plot showing how weeds can reduce yield. Anything? Something happened?
I'm just going to close this box.
Oh, okay. Cool. So this is a plot showing how weeds can reduce yield. The black gray bar represents the potential loss if we don't do anything to manage weeds, but then we can reduce that bar so that losses are prevented, and we only reduce yield by 10% because we do a good job controlling weeds. They can reduce the marketability of the product in case, again, I was talking about weeds in seed crops. If you have a seed lot for annual ryegrass that will be used for cover crop, and if you have some annual ryegrass with resistance to a herbicide, say glyphosate, you're going to lose a lot of the price for that cover crop seed lot, because how can you terminate annual ryegrass with glyphosate if you have infestation or contamination with herbicide resistant annual ryegrass? Loss of land value. If you have, if you're looking to buy a piece of land and you have two areas to choose from, one is infested with herbicide resistant weeds and the other one is free, which one are you going to pick? They can serve as alternative hosts for other pests. And most of the weed management in the United States are conventional, meaning they will use herbicides to manage weeds. And this is a problem because herbicides work so well when we end up over relying on them, and that leads to herbicide resistance. So herbicide resistance is something that it's not new. If we see in this plot here, we had the first report of resistance in the 60s. So since then, we have over 500 cases of herbicide resistance around the world. This is a plot showing numbers in the United States, Europe, Australia, Brazil, Canada, and so on. So the United States is the country that has where most weeds evolved resistance, and they make weed management much harder. And out of the 31 mode of action that we have nowadays available for growers and farmers to spray, weeds evolve resistance to at least 21 of them. Okay, so how does herbicide resistance happen? It's a consequence of naturally occurring mutations. So the mutations are already in the population. So if you think about a population that has most of the individuals will be susceptible, they are represented by the green plants here, and then a few of them in low frequency, they have the resistance allele, the resistance variants that will confer resistance to this herbicide being sprayed. If we spray this herbicide one time, because weeds can reproduce, they will increase in frequency. So in the second year, they're going to see much more resistant individuals. And then by the third year, if you spray that herbicide again, you're going to have more weeds because, again, they increased in frequency in the population. Okay. So most of the numbers I showed you, at least in the United States, they are driven by weeds that occur in soybeans. So if you look at the total numbers of reports, and these I'm looking at number of just regular, reports in general, not only unique. When we're talking about unique, we are only thinking about one weed that evolved resistant to one herbicide. And if that happens multiple times, we would still consider it as unique. In this case, is how many reports there's being out there. And so when you look at the percentage, the proportion of herbicide resistant weeds in the United States, most of them are in soybeans, much driven by resistance to glyphosate. ALS inhibitors, it's just a type of mode of action, ALS. ALS is actually an enzyme that a group of herbicides inhibit. PPO or Protox is another group of herbicides that some herbicides will, PPO is an enzyme, and then there are the PPO inhibitors that will inhibit an enzyme and kill the weeds, and some other types of herbicide resist as well. Kind of a heat map, and so far I'm just showing you how big the problem is of herbicide resistance, with more focus on the United States. This is a heat map showing the different states in the country, and what the red ones represent the most herbicide resistance, and the blue ones, not as many reports. So we see, for example, California would be a state that reported many cases of herbicide resistance, Kansas, Illinois, Arkansas, some other states, versus other states with a colder color like New York and Pennsylvania. So a few things that take away message from this map and we should think about is we need to be careful when interpreting this plot because, for example, if you think about Kansas, the fact that it has many weeds reported doesn't mean that all of the weeds are problematic. It means that there are very good people like Vipan here was doing really good job at reporting and finding and doing the research. So, if you have a state, for example, in Pennsylvania, where you likely have more resistance, but you don't have a weed scientist dedicated until I started there to really look at herbicide resistance as the main focus of the program. So it's more likely that there are more herbicide resistance cases that could be reported, but that haven't been reported. And another thing to be careful when interpreting this map is, for example, some other states that have not as many weeds reported, for example, Colorado, that doesn't mean they don't have problems with herbicide-resistant weeds. In fact, they do have, but maybe one plant is really problematic. For example, they have kochia, which is a broadleaf weed that is very problematic throughout the state. But in general, this map kind of has a strong correlation with how problematic the weeds are in each different state. When you look at broadleaf weeds versus grasses, you're going to see here, and I apologize for just skipping the Latin names, but the weeds that have evolved most resistance are broadleaf weeds, or most reported. Palmer amaranth is a type of pigweed. Water hemp, number 2, is another pigweed species. Kochia scoparia, another broadleaf. It's called kochia. This is horseweed or mare's tail, depending on who you're talking to. Different common names, but Conisa canadensis. And then we have Lolium multiflorum, which is ryegrass. I'm going to talk a little bit more about later in this presentation. So we have all this resistance is widespread. Usually when we talk about herbicide resistance, there's a rule of thumb that we use to consider a weed population as resistant. Usually it's a survival to 2x the field rate. But it varies, right? So the fact that we have resistance doesn't mean they have all 2x or all have 100x or 1000x, and really depends on how the weed is dealing with the herbicide. So a couple of examples here. When you have a weed that is resistant to ALS inhibitors, usually resistance is really high. So weeds can survive to up to 1,000 times what a farmer would spray in the field. And then glyphosate resistance. Glyphosate is another super important herbicide nowadays. It's the trade name is Roundup and many different versions. Some weeds can survive to just 2 times the field rate, but some much more than that. So it really varies. And how much a weed can survive to a herbicide is driven by the resistance mechanism. Okay, so weed scientists, we divide resistance mechanisms as target site and non-target site. So when we're talking about the target site, that it's kind of a silly classification. Target site is when there are changes to the target site of the herbicide. So this is just a quick example here showing an enzyme that the herbicide targets. This kind of a mushroom icon is the enzyme. So a resistant weed evolved resistance, so the herbicide no longer can inhibit that enzyme. Another way that weeds can evolve resistance is by overexpressing the genes or the target site. So instead of having just a few molecules of the herbicide inhibiting the yellow ones that are representing here the enzyme, you have way more enzyme and not enough herbicide to inhibit all the enzyme. So basically overexpression of the target site. And then the non-target site type of resistance, we have many different types. I'm just summarizing a few of them. One type would be a kind of reduced translocation, so the herbicide doesn't move as much. and that can be because of a cooler sequestration of the herbicide. So the herbicide gets pumped out of where it's supposed to go. Typically, herbicides will need to go to the target site. They are located in the cytoplasm, in the endoplasmic reticulum and chloroplast, but instead of going those locations, the herbicide gets pumped into the vacuole and away from the target site. There are some exclusion mechanisms as well, pumping out the herbicide from the cell. And then there are also the metabolism of the herbicide. So basically, the plants, the resistant plants, can break down the herbicide, so the herbicide doesn't have that inhibitory characteristic. Okay, so now you have a better idea of how herbicide resistance happens, some of the mechanisms, how important herbicide is, and how widespread it is in the United States. So now moving on to annual ryegrass. So I'm a little bit biased because I chose annual ryegrass is a model species for me. My program has been using annual ryegrass for quite a few years to try to understand resistance. So moving on, I will focus on this species to show some of the projects going on in my lab to try to tackle and better understand the herbicide resistance. So annual ryegrass is a winter annual species. It's an obligate outcrossing. That just means that to produce viable seeds, one genotype needs to mate with a different, exchange genetic material with a different genotype. And overall, in the world, there's been 73 populations with herbicide resistance to annual ryegrass. If you account Lolium rigidum, rigid ryegrass is a close relative to annual ryegrass. Oftentimes they are misidentified to each other, so it's sometimes it's hard to tell them apart. But if you consider both species, there's been over 120 reports of resistance in this species. If you compare Palmer amaranth, there's been 74, water hemp 64, and horseweed or Meristeo 67. So again, it's a weed species that evolved resistance to many herbicides multiple times. I should emphasize that's multiple times. Common resistance in ryegrass would be resistant to glyphosate, ACCase inhibitors. ACCase, again, is just a different type of mode of action. So ACCase is an enzyme that is involved in the lipid biosynthesis. And ALS is an enzyme involved in the synthesis of amino acids. So basically, resistance to those modes of action. There is a population I've worked on when I was a grad student, and it had resistance to a single population resistance to 4 modes of action. And we do know of populations of Lolium rigidum, rigid ryegrass, again, close relative to annual ryegrass, with resistance to 7 modes of action. So again, because of all those things, it's a weed that can be a very good model to understand herbicide resistance evolution. Okay, so where is ryegrass a problem? So these I'm showing 3 pictures, each one in a different location. One on the left-hand side here, it's a clover field. In Oregon, white clover in this case is grown for seed. So it's used for forage, cover crop, and so on. And you're going to see, I hope you can see a lot of grasses. This is ryegrass. It's primarily managed with herbicides. Growers will, and this is the Oregon, I don't know if I already told you, and growers will use a lot of the ACCase inhibitors, such as clethodim and fluazifop, but mainly clethodim for control of Italian ryegrass. In wheat, ryegrass is also a problem. This is a picture in the middle. Again, there are a few ways to control ryegrass in wheat. One of the main herbicides would be pinoxidin, and ryegrass on the right-hand side here, infesting a prune orchard in California. That's where I had my field trials for my grad studies. This population in particular was just treated when this picture was taken with paraquat a couple of days, or I think a week before. And paraquat is supposed to work very quickly, very fast. You can see plants dying from paraquat treatment 2 days after treatment. So this is another location where Italian ryegrass can happen. And as you can see, Italian ryegrass is primarily managed with herbicides. This is the primary way of controlling these species, regardless of what cropping system you are talking about. Okay, so again, another reason why we see so much resistance in ryegrass, it's because we use so much herbicide to control it. So how does ryegrass do it? One of the advantages that ryegrass has is that ryegrass has a very large genetic diversity. So this is a study that we completed recently. We just submitted a paper looking at a specific allele that gives resistance to ryegrass, to a specific herbicide. I'm looking here at the ACCase allele. And we screened, we sequenced this allele in almost 100 plants. And out of the 100 plants that we sequenced, very few of the alleles were unique. Basically, we had a lot of genetic diversity, a lot of differences, mutations in this specific gene. So that's just to show you the size of the genetic diversity of this species. So the more genetic diversity species has, or the different populations have, more options that evolution can kind of pick from and then give rise to resistant plants, okay? Another thing that we observed was a very strong admixture. So admixture just basically means populations from one location exchanging genetic material with populations from a different location. So a lot of movement of alleles in ryegrass. And that makes sense when you think about it, because ryegrass is a very, it's a weed that can survive many different places, locations, climate, stresses, and it's a plant that is used for cover crop, for pasture. So we have a lot of breeding programs where ryegrass is being bred for different characteristics. For example, there are research programs looking at breeding ryegrass in New Zealand, in Uruguay, in Oregon, and then they are always selling the seed to different places around the world for cover crop, pasture. So there's this mixture facilitated by just how agriculture works, movement of seeds and genetic material everywhere. And how does that help? How does that help with evolution? So if you think about a population that has a pool of alleles, so the diversity here is represented by different colors, and then you have environmental stress. That environmental stress, that could be a herbicide application, will reduce the diversity in the population. So you see a decrease in the kind of number of colors here. And then that population will become much less diverse. But then because of this movement of alleles, there can be new reintroduction of diversity. So that's how ryegrass, we think there's a kind of a theory that we have in terms of for ryegrass, that it can rebuild genetic diversity because of this movement of alleles and genetic diversity facilitated by agriculture. But much remains unknown when it comes to non-target site resistance. So we don't know, we do know a lot about target site resistance. We usually know what the target site of a herbicide is, but we don't know how herbicide non-target site resistance works. So I'm giving you here a couple of examples. How much time do I have?
Left over.
What time am I supposed to finish?
Around 12:05.
12, okay, cool, got time, thanks. So non-target site resistance, we don't know a lot about, but I'm gonna show you a couple of examples and what we are trying to do to elucidate more when it comes to non-target site resistance. So this is an example showing a paper we published some time ago on paraquat resistance. Paraquat targets the a pathway in the chloroplast, so it's supposed to go here. So it needs to enter the cell. And then what we think is happening is that paraquat in this resistant population of ryegrass, paraquat can either be pumped by the cell into the vacuole away from the target site, which is a chloroplast, or pumped out of the cell by a plasma membrane bound protein that we are still trying to figure out what happens, which one it is. So we have some visuals here as well. Sorry about that. So you have a treatment here only with a inhibitor of the protein that moves paraquat around. You don't see any damage to the plants. And then you have only paraquat. This is again paraquat resistant. We don't see much damage to those to the plant. And then when we pre-treat that inhibitor of the protein, and then we treat paraquat, then we can reverse the resistance. So that's how we concluded. We think that's what's happening in this population. But again, we don't know what are the resistant mechanisms, really the underlying basis of the resistance trait. We don't know the protein, we don't know the genes, and we don't know what happened to the genes. Is it something that's happening in terms of changing expression, a promoter, mutation in the promoter region, and it's single nucleotide polymorphisms. Many things can happen that will change how a protein can work in the plant. This is another example, again, in ryegrass, showing how population evolved resistance to glufosinate. We see that this population can metabolize glufosinate. Glufosinate is, again, I don't think I talked about it. Glufosinate is a herbicide that's very popular. It's you might know it as Rely or Liberty. You have all the Liberty Link traits in different crops. That gives resistance to this herbicide. So again, it's an important herbicide. So this population evolved resistance to glufosinate by metabolizing it. But again, we don't know what are the underlying bases of the resistance trait. So it's still much to be discovered about non-target site resistance. And I'm emphasizing here on non-target site resistance because it can be very difficult to deal with weeds that have non-target site resistance mechanisms, especially when weeds can metabolize the herbicide, because herbicide metabolism can be non-specific. So this is a very cool research looking at a cytochrome P450 enzymes. They are known to metabolize xenobiotics, basically herbicides. Humans have P450s that can metabolize different things, different molecules in your body. So in this case, if you focus just as an example on this cytochrome 81A24, we see that if a plant has more activity of that specific cytochrome P450, it will have resistance to mesotrione, which is HPPD inhibitor, a mode of action, also to PDS, another mode of action, PPO, another mode of action. So one change, one overexpression of one enzyme will give resistance to many different herbicides, and that can be really problematic for farmers to manage this type of weed. But again, we don't know what happened. We know the gene. In this case, it's really cool because we know the gene, but we don't know what happened to that gene. Why is it overexpressing? There's still much to understand about metabolism, herbicide metabolism in weeds. And beyond, again, along the same lines, herbicide metabolism can be problematic because it can give resistance to herbicides that haven't even been used yet. So this is a very cool example from Hudson Takano and collaborators. They were looking at a herbicide called fluoroperoxyfen. It's a rice herbicide, and they studied two populations denominated AR27 and MO18, and they saw that these populations are resistant to imazamox. It's a herbicide that inhibits the ALS. It's a mode of action. And then when they sprayed this other mode of action, fluoroperoxyfen, they saw those populations were also resistant to fluoroperoxyfen. So basically, fluoroperoxyfen is a new herbicide never being used in rice. So it evolved resistance to imazamox, and because of that mechanism of resistance, it can also survive to fluoroperoxyfen. That can be really challenging for everybody, for growers who are using a new herbicide, for the chemical industry they are developing new chemicals, but they have that metabolism. They will launch a new herbicide and it might not work because some weeds can already be resistant to that herbicide. In this case, again, we don't know whether they are the herbicide resistance mechanisms are and what are the underlying bases, what are the genes, what are what happened to the genetic code in this case. So how is my program trying to address some of that? So I think there are many opportunities if we start to understand better the genetic codes of weeds. If we understand and have resources to like genomic resources specifically, we can start asking questions such as what are the genes involved, what happened to the genes, what happened to the genetic code that gives now resistance to a weed, how herbicide resistant individuals move, resilience mechanisms. We know some weeds can be very plastic and survive to different conditions. I mean, everybody who saw a weed or tried to manage a weed in a field will see that they have better adaptation to hot climates, to changing condition, drought, heat, and all of that. Compaction, and other mechanisms of adaptation as well. So if, in addition to understanding herbicide resistance, genomic resources in weed can also give us more tools to understand how weeds do that, how they are resilient. And perhaps in the future, we can use those traits to breed better crops. So if we know what are the mechanisms for plasticity in weeds, why not take those mechanisms and incorporate into a new line, a variety of a crop that we are interested in growing? So the first thing that we did specifically for ryegrass, Indian ryegrass, was to assemble a genome. Basically what it means is to sequence all the nucleotides in the genome. Usually the tools that we do that, it gets very fragmented set of sequences, and then we use computers to assemble the genome and try to put together those pieces of DNA and get a linear, longer scaffolds that we can call eventually chromosomes. So in this case, we did a pretty okay job. Lowly is supposed to have 7 chromosomes. We're able to assemble over 95% of the reads in a single in 7 scaffolds, so it's a pretty decent assembly. So that's the first step in trying to understand the genetic code and use that as a resource to ask some of the questions that we have about resistance. And I wanted to focus on paraquat resistance as one of the first questions that we ask, because one, it's something that my program already has some experience on. We had also all the populations that we could use to try to understand it. So basically, we tried 2 different approaches. One is called a genome-wide association study. Basically, we take resistant and susceptible plants, and then we try to associate some of the variants in each population and see if they are associated with the resistance phenotype. Basically, this is a process of discovering markers throughout the genome of the weed for both resistant and susceptible. Very quick summary. And then we also did what's called a QTL-seq, a quantitative trait loci analysis. We took two different parents, one resistant and one susceptible, paraquat resistant, paraquat susceptible, crossed them and then crossed them again. And hopefully what we're looking for were regions of the genome that could be associated and segregated along with the resistant phenotype. Okay, so a little bit more details. We took many field populations, and some of them were resistant, some were susceptible to paraquat. We sprayed them with paraquat. We collected DNA, extracted, and did what I just told you we did about the markers. We discovered markers throughout the genome, thousands and thousands of those markers. And for the second approach, we did what's called a pseudo F2 cross. Without much details, eventually we obtained a population that's segregated for resistant and susceptible. You spray them and collect the DNA, and we sent for sequence. This is something that usually people will do that for you. It's a service for fee. So here are some of the results. I'm showing here the results first for the GWAS experiment. You're going to see, if you're familiar with this type of plot, this is a Manhattan plot. It's called Manhattan because it looks like a bunch of buildings put next to each other. And the numbers down here are the scaffolds, basically the fragments of the genome that were contiguous. This is a little bit more fragmented because we did this analysis some time ago, a few months ago. We have a better version of the genome that we are going to redo the analysis, but I don't expect this data to change much. But we did find, so whenever so each data point here represents one marker that we had in the data. So in this case, I can remember we had close to 1 million markers throughout the genome. And the data points that fall above this line represents they are associated. The location where they are, it's associated with the phenotype. In this case, is resistance to paraquat. So we use two different models. They agree, and we see a couple of regions of genome that were on scaffold 8 and 44. I don't want to bore you too much with the details, but keep that in mind, the 8 and 44, when we did the analysis with the QTL-seq approach, we also found a couple of hits in scaffold 4 and 44 and 8. When we looked more closely to those markers and regions, we found there were a few ABC transporters that were located in that region. So basically sequences of DNA that encoded ABC transporters. And we know that ABC transporters are involved with resistance to herbicides and resistance to drugs in other species. So again, it's kind of false, kind of agree with previous data that we had from a few years ago that ABC transporters might be involved with paraquat resistance, because those in particular that I found are located in the plasma membrane specifically. So it's likely that they are involved, and we're going to do some more characterization of those. So these are some of the things that we're doing to further our understanding in terms of non-target site resistance. Okay, so we found a bunch of sequences. We tried to understand. We found the genes that give resistance to paraquat. We can use that same approach to understand resistance to glyphosate, to ALS inhibitors and CCase inhibitors, PPO, all the resistance. We have the potential to try to further our understanding. But what we can do in terms of more applicable, practical things that we can do with that information. So anyone working weeds or that see a weed surviving in the field, the first question that we ask is this really herbicide resistance? This is a question that we ask because many other things can happen in the field that will reduce the efficacy of a herbicide. There can be climatic conditions. There can be a quick rainfall after application. The nozzle can be clogged. There can be shading, one leaf shading the other, and you get a poor control. The size of weeds might be different. The soil might be too heavy and you spray a pre-emergence herbicide. So many things that can happen in the field. The plant can be stressed, heat stress, drought stress, and so many things. So how we do that today to make sure we have a case of herbicide resistance? The typical way that we do this, we characterize resistance, is by doing a dose response. That basically means you take the population that you think is resistant and compare that to a susceptible plant. And then you spray different rates and you see, you build the dose response models here, basically correlating the two and seeing what we call the resistance ratio. So basically this is what we do, but if you think about it, we need to spend time germinating the seed. Some seeds might have dormancy. They can need to be stratified or something that will delay germination. They can be grown. They can get to a decent stage to spray. Then you need to wait for the herbicide to work. That can be very fast for paraquat, a couple of days. It can be very much longer. For glyphosate, for instance, you need to wait 2-3 weeks to get definite results. So basically the whole thing takes 2-3 months. So that's a very, very long process of understanding resistance. And ideally, this should be quick, because what we want to do is to try to adequate our resistance management actions within field and not wait until the next year to do that. You don't want to know if you have resistance next year. You want to know today if you have resistance or this week. So with that information, and this is a plot from one results from one of my students is working on, Luis. He knows the resistance mechanism of a particular weed species, and what he did was to develop a quick assay to detect resistance. Basically, this is a discrimination plot. Whenever a weed is resistant, the genotype will fall in this region here, represented by the red ones. Or susceptible in the blue ones here, and if they are hybrids, it would be kind of in the middle. So this is a lab type of experiment with the caveat that we need to know what the resistance mechanism is. The beauty of this is that you can use that for a leaf sample. So you need to collect this leaf in the field, bring it to the lab, extract DNA. The whole process, takes probably a couple hours to check DNA, and then another 40 minutes to run this assay. So you can get results. If you have the leaf tissue, you can get results in less than 3 hours, more or less 3 hours. So that's quick enough, but it can be quicker. I'm going to show you later. And this can also be used for seed lots as well. So if you have a seed that you want to know if it's contaminated with resistant weeds, you could use this type of assay to see if you have resistance or not. And then you can have this more lateral flow, kind of a COVID assay that we do all the time, where you just rub a little bit of the leaf, put in the lateral flow device, and then it tells you whether you have resistance or not. This is more on the long-term development, but there is technology out there that can we can develop this type of assay. And some other more of the genetic control weeds. You had Todd Gaines coming here last year to talk about some of his projects on this area, but using RNAi technology to kind of reverse resistance. That's another possibility. But the first thing that we need is to know what the resistance mechanisms are, what regions are involved with the resistance, and so on. Okay. And if we know the resistance mechanism, we can start anticipating what to do. If we know the genotype of weed in the field very quickly, and we know what genes can give resistance to multiple herbicides, if we know that information, we can better choose the management practices that we use to control resistance and kind of anticipate what to do in the field. Okay, so very quickly here, I'm running out of time, but some other opportunities beyond using genetics. There's this really nice research by Andrew Knies and collaborators looking at the cost of using different herbicides, ideally for integrated weed management. It's recommended that multiple herbicides are used either in rotation, but the preference is given to mixtures. So, but the problem is that mixing herbicides can be costly. It can cost money because you are proactively adding a chemical that will cost more money. So one solution to this is another really nice work by Hudson Takano looking at true synergistic mixtures. So you have in this plot showing the action of glufosinate and visual control. The bigger the bar, the better. So you have glufosinate here only giving about 25% control. If they mix a very small amount of a herbicide that really synergizes with glufosinate, and 1 gram in this case, it's like 5% of the field rate is the field rate divided by 20. So that will give a much better result, almost to 100%. So identifying those optimal mixtures that synergize herbicides can be another way, more on the prevention of herbicide resistance instead of relying only one chemical. And we can also improve our understanding of pre-emergence herbicides. Pre-emergent herbicides, they are a little bit more challenging compared to post-emergent herbicide, in my opinion, because there are so many things that are happening. There's a lot of things happening for post-emergent herbicides, but for pre, there are way more going on in the soil. You have all the soil characteristics, the microbiota, and so on. So this plot is showing you the herbicide efficacy, the herbicide concentration in the soil. And two herbicides that were sprayed, one blue line and the other one red. And whenever this line falls, the concentration falls below the efficacy threshold, that just means the herbicide will not be killing the weed anymore. So if you use a mix of 2 herbicides, and that's usually what we recommend, mixing herbicides, you are going to see that the blue line here, the blue herbicide, will fall below the line much quicker than the second one. This first one only has a residual activity of 1 month versus the second one over 2 months. So this interval here, only herbicide B, the red one, is working. And that's giving selection pressure as the grower had just sprayed 1 herbicide. So understanding those dynamics can give us much better management practices that will reduce selection pressure and evolution of resistance. So in summary, we need to better understand herbicide resistance evolution, particularly because with that knowledge, we can better manage and integrate integrated weed management practices in our programs. Quick diagnosis, understanding resistance patterns, what herbicides will work, as well as better mixtures of herbicides. And we really need to better use the chemicals that we have available today, because we haven't had any new herbicide in over 30 years, new mode of action in over 30 years. So we really need to do our best to preserve the tools that we have nowadays. And, just what I reemphasize what I already told you, quick identification important, prediction of cross and multiple resistance, optimizing take mix partners, and other new, and I didn't talk about this, but there are folks here like Linus Sosnowski doing a great job with new and emerging technologies for weed management, vision guided spray, robotics, electro weeding. Those are things that should also be incorporated in managing herbicide resistance proactively and reactively after they are evolved. Cool. With that, thank you so much for your attention, and happy to answer any questions.
Okay, great. That was excellent. So, yeah, I think we've got plenty of time for questions. And if you're online, feel free to unmute. If you're in the room, feel free to ask a question. And also, if you're online, if that would be easier for you, feel free to put your question into the chat.
How many folks we have online?
Thank you. So,
We had up to
Quite a few.
32 online.
Yeah, cool.
Yeah.
We saw.
thank you so much. My name is Cedric Asoba. I'm from Benin, West Africa, and thank you for this nice presentation. It was a wonderful one. I really appreciate everything you said. And I just want to ask a question. I work on herbicide resistance, mainly on the molecular mechanics of herbicide resistance in Lolium residuum. And finally, it was based on the targeted resistance. We identified different mutations, but after molecular docking studies and also secondary structure studies, we didn't find any difference, any change in the protein structure. And I was wondering what could be the reason behind that? Because after the screening, the bioassay analysis, we got differences, we got changes, and we clearly noticed that there is a change in the resistant biotype, like they are resistant, effectively resistant, but the susceptible ones, they are not resistant. And I couldn't explain that. I don't know if you have any idea or any suggestion for that.
So what mechanism of resistance you're talking about? Let's see if I got the sound in the room is not great. So basically, you're talking about ACCase inhibitor resistance.
Exactly, ACCase inhibitor resistance, and I use pinostatin as herbicide.
Okay, you have target sites. So when you have different type of target sites, you don't see much changes in the resistance factor. Is that the question?
Yes, like we got different mutations, but after getting the mutations, we try to perform the molecular docking to see the protein structure and how it evolved, because we know that in case of target site, there should be an interaction between the active site and the protein, the enzyme, and so on. And we didn't get any change. I couldn't explain what is the reason behind that.
So you don't see any changes in the interaction between the herbicide and the active site of the enzyme. Is that right?
Exactly.
I don't know. That's a good question. So that could be, I don't know. I'll have to think about it. So basically in your modeling, you did not see any changes in how the herbicide interacts with the enzyme. Okay, I'll get your name and then think about it.
Okay, thank you so much.
Yeah, we can even talk later if you want to send me an email.
Okay, thank you so much.
That's a good question.
My name is Cedric.
Yeah, Vipa.
Good presentation, Kyle.
Thank you.
We enjoyed it. You mentioned about the ryegrass as a model species for understanding the genetics and the mechanism of resistance. That's a grass species. How about broadleaf species? Do you have anything in mind? What will be the best model species to understand these things?
Yeah, Vipa's question was basically ryegrass I'm using as a model. What about broadleaf weeds? You know, you need to really think carefully when I say model, because it's a model species. So there are other things that should be considered when you think about a model species. As you know, if I talked about this to other plant biologists who are working in Arabidopsis or Bromus or Brachypodium, green foxtail, all those actual model species, they would laugh at me, because Lolium doesn't have much in terms of model species. It's a outcrossing species. It's it has a lot of repetitive elements in the genome. The genome is huge. It's like 10 times bigger, more than 10 times bigger than the one of Arabidopsis. So, but model, I say because it has a lot of resistance. Another one that would be super excited about trying to work and further understand it would be Palmer, Palmer amaranth, water hemp, those already, there are genetic resources in those species. So I think, and there are really good research people working on those species. But usually, you know, I don't know if I used really well the word model, but I do like to incorporate, to do research on things that will be impactful and have relevance to management. So that's why ryegrass was the one that I wanted to focus on, because it's so problematic and we needed to better understand what's going on. Does that make sense? Yeah.
Follow-up question.
Yeah.
You mentioned about paraquat on annual ryegrass. But paraquat generally doesn't work that good on grasses, right?
Yeah.
So was it doing well for annual ryegrass in your?
So usually, yeah. So paraquat doesn't work well when the weed is that big, because it's just a burn down herbicide. It's a contact. But you know, in that picture that I showed you, we would expect a good burning of the leaves. But then when we bring the weeds to the greenhouse, we spray them when they are 2 tillers, and paraquat controls well the weed. It's pretty good. In perennial crops, paraquat is still used a lot. It's a good alternative to a number of species, like even horseweed, marishtale growers use a lot because glyphosate doesn't work anymore. It's hard to find a population that's susceptible to glyphosate in horseweed. So paraquat still works. Most of the time, Lynn Sosnowski found a lot of populations here in the state, in New York, that are resistant to paraquat. So that's another challenge. But in general, paraquat works well.
And also you mentioned about I think I have more questions.
Yeah, please.
You mentioned about the transporters involvement in resistance to paraquat.
Mhm.
How about the other non-target sites mechanisms like metabolism or GST? Are you characterizing those genes or?
Yeah. So basically the question is, what, in addition to the transporters for paraquat resistance, what about the other ones? What are mechanisms like metabolism? So in that paper I showed with the little scintillation vials, we also did HPLC analysis, and we didn't see any metabolites. So paraquat is known to not be metabolized in plants. So I don't think metabolism would be involved. But we did see difference in translocation, how much moves in the plant. So that's why it makes sense to be a transporter. But for other herbicides like glufosinate, I'm also working on that as well. So I have another, I didn't show here, already have some data, but basically we did a lot of the QTL, we did another QTL analysis, RNA-seq. So we have a few ideas what might be going on. Yeah.
Great, thank you.
Lynn's got her hand.
Great, go ahead, Lynn.
Oh, hey, Kyle, that was awesome.
Thanks, Lynn.
Thanks so much. I was wondering if you could talk maybe a little bit about temperature impacts on resistance, or at least the expression of resistance, and how I know you know environmental conditions can shift responses that we see, and maybe what that means for changing climate.
Yeah, that's a really good question. Thanks, Lynn. Specifically for paraquat, and I'm familiar because I did the experiment, when you have a lower temperature, about 16 Celsius, which is 70 more or less, I would say, or even a little bit lower, paraquat doesn't work as well at lower temperatures. But if you increase that temperature, paraquat will work better. And I think that's the same trend for glyphosate. A higher temperature will, or maybe for glyphosate it's the opposite. But in summary, temperatures will affect how much you get in terms of efficacy of a herbicide. And each herbicide will have one different, trend that we will follow. Beyond environmental conditions after application, we can also think about environmental conditions before application that the plant has been experiencing over the years. There's one thing that we call the priming or basically pre-acclimation of the population to environmental for stress. So we do know from other species that previous express can be transgenerational. Basically, if you give the plant heat stress, for instance, there will be a lot of reactive oxygen species amplification and damage and drought and so on. And then you spray a herbicide that will kill the plant by enhancing the ROS and all of that. It's possible. We don't have good research yet that I'm aware of showing that relationship, but it's possible that the environmental stress will pre-acclimate the plant to deal better with herbicide, and that can be more important moving forward with climate change, now stronger than ever, but getting worse.
Okay, I think we're just about out of time, so thank you, everyone.
Cool, thank you.
And,
Thanks for your attention.
Thank you, Kyle, for the excellent presentation. And, thanks, everyone online.
Thank you.